Okay, so let's talk about the scientific method for just a minute. So we covered that in class, and I want to make it clear that, you know, a lot of times the book or when you're talking about the scientific method, it's all about there's this step and then there's this step and what is that step called and what is this step called. And it is a step-by-step and it's important, but there's some key things. It's the order in which these steps happen that's important. And so, you know, at the very beginning, there's got to be something that triggers us to start thinking about something, right? So at the very beginning, we're going to have some kind of observation that occurs, right, that triggers us to think for a second. So could be a question we have, something interesting that we've observed. So the example that I'm going to use today has to do with tomato plants. So I've tried growing my own tomatoes quite a bit here in Texas, and I've not had a lot of great success. So let's say that I make an observation that my neighbor over here seems to be able to have success growing tomato plants, and I seem not to be able to do quite as well. Well, obviously we're located in the same general spot. You know, let's say that the sunlight and the soil and all those things are pretty comparable. So I begin to observe what's different about their tomato plants than mine. And maybe they tell me, oh, I use this particular special plant food. And that's the trick. That's what makes my tomatoes taste better, grow better, whatever. So then I move to the next step. So as part of the scientific method, then I'm thinking, okay, could that be it? And if so, how would I be able to find that out? And so this would be where we would start to develop a hypothesis. Okay, and the key thing to a hypothesis is it must be testable. So another way you could say that is it must be, I don't think I'm going to be able to fit this, falsifiable. I don't even know if I spelled that correct. You must be able to disprove this, okay? In some way you have to be able to test whether this could be true or not. So let's say that I develop a hypothesis that says tomato plants grown using Miracle-Gro plant food will yield a better crop than those grown without plant food or plant food by another brand. Okay, so then I need to figure out how am I going to design my experiment, right? So that, let me spell this, experiment. Okay, I'm going to design my experiment that will in fact test the hypothesis. Now this right here, experiment, so this is key, right, to the scientific method. This is what makes it so reliable, because I don't just guess, right? I don't just come up with possible solutions. I'm actually testing whether or not this could be accurate or not. So that's the next step. Okay, then we're going to collect data, and finally we're going to draw some conclusions about what does that data say. Does it support my hypothesis or does it reject my hypothesis? So that's the overall way that we look at the scientific method. So here would be my question, and that would be, if I'm going to design an experiment, there's several things that I have to think about in order for it to be valid, right, to test what I actually want to test and not something else. Okay, so we've already come up with a pretty good hypothesis, right, for this. And it's important that in our experiment we recognize that there's going to be some standard variables that we want to be held constant. So these are things that we don't want to influence our test. Okay, so if you think about, in this case, if you think for just a second, we could come up with several standard variables that we would not want to be different in our test subject. So for example, I want the amount of sunlight that each test plant is getting to be the same. Okay, I want the water, the time of day, the amount of water that I give, all of those things should be constant, right, between all my plant groups. And also the variety of my plant, you know, the age of it, the health of it before we got started. You could go on and on, right? These are standard variables. We want these to be the same so that they don't influence our experiment. Now, I do want us to talk for a second about the independent variable and the dependent variable. So the independent one is the one that as a researcher you are manipulating. Right? This is the one that you are affecting. The dependent variable depends on the independent. So in this case, let's, you know, if we in fact are looking at, so Miracle-Gro would be one, right? That would be one group. The Vigoro brand would be another test group. And the Shake and Feed brand would be a third test group. And then I'm going to need a fourth test group, right? Which is going to be my negative control. So this one is going to get everything that the other three groups get. Same water, same sun, same plant, but I'm not going to give it any plant food. So we're going to set up the experiment with at least these 4 plant groups. Now, the other thing about designing an experiment is the more subjects that I have, right, the more plants. So if I can repeat this, so in the Miracle-Gro group, if I have 10 plants versus just 1, right, that's going to be a lot more meaningful than if I just have 1 plant. Okay, so get back to the independent variable. Whether I'm using a plant food and what brand, right, that's going to be the independent variable. So am I using Miracle-Gro? Am I using Vigoro? Am I using Shake and Feed? Or am I using none? That is my independent variable. What kind of plant food, if any, does it receive? Now the dependent variable is something that we would have to determine. So it kind of depends on what we consider more successful as far as tomato plants. So the number of tomatoes that we produce, the size of the tomatoes, how the tomatoes taste, how healthy the plant is. We would have to have certain things that we define as, this is what we mean when we say better tomato plant. So those things would be the dependent variable. Okay, so we're measuring those to say which one is yielding the better tomato crop. So size of tomato, number of tomatoes, so on and so forth. So that's sort of scientific method. And I just want to also point out before we move on to another topic that there's a difference between pure science and applied science. So pure science is going to be when you don't necessarily have a specific problem in mind that you're going to solve. So you may be studying it to truly to just discover something. So in academia, at universities, there's pure science conducted all the time, and many times that leads to applied science because those things are developed to do something or to help in some way. But applied science would have a specific goal of, you know, solving a problem or fixing something. So that's sort of the difference between pure and applied science. So I'm going to move on. We're still within lesson 1. And in lesson 1, we talked a little bit about living things. And one of the things we talked about was that living things have certain characteristics. There we go. That are shared among all living things. And one of those characteristics is that they all have some kind of organization. And this down here is just a graphic that shows how, as we go from atoms, so the very smallest atom, all the way up to ultimately the biosphere, that all the levels in between, that there is some kind of functionality that is gained as we move up in complexity. Now, I just want you to think for a second about which, along this graphic, see if we can fill in what's missing. So atoms, molecules, cells, and next would come, let me change my color here, tissue. So when we have a group of cells working collectively, right, then that forms a tissue. Now, if we have a group of tissues coming together to perform a specific function, then we get an organ. And if we have multiple organs working together to perform a related function, so that might be like the bladder and the kidneys and the ureters and the urethra. So that would be an organ system. And if you think about, a cell, right? A cell has a certain functionality. A tissue, right, can do more than a cell. An organ then, for example, the bladder, right? It has the ability to stretch and contain urine. But the organ system, further, the bladder, kidney, it has the ability to filter the blood, store the urine, pass the urine to outside the body. So we have a gain of function. And then organism, when we move from one organism to more than one, the first category is going to be population. And a population is going to be one species, okay, that can interbreed. So a population of, I've got a population of frogs that live sort of near my house. A population of a specific species of birds. So I have multiple species of birds that will come to my backyard. So each one of those species would represent a population. Now, a community is all the living things in a given area. So the population is also in a given area, right? But it's one species. So community would be all of that. So if I'm talking about my backyard, or my neighborhood, then all the birds, all the frogs, all the insects, all the fungi, everything that's living, wasps, bees, all that goes into the community. And then we have a step between community and biosphere, and that is ecosystem. So what does the ecosystem have that the community doesn't? Well, we're going to add in what we call non-living. And sometimes it's referred to abiotic. So biotic would mean living, right, life. So A is without. So the non-living abiotic things, for example, water, air, rocks, soil, that kind of thing would be part of the ecosystem. Now, all the living things are part of it too. We just add in the non-living component. And the biosphere is essentially the entire surface of the Earth that has anything living existing on it. Now, before I move on to the next lesson, I do want to point out that I do want you to be familiar with the characteristics of life, and specifically examples of those characteristics of life. So let's see if we can name a few of these and maybe give some ideas of examples of each of those. So we talked about order or organization, right, that we just went through. We can list some others. So let's say respond to the environment. Okay, that one's pretty easy to see some examples of those. So for example, if you think about in nature, if a predator, right, if the prey sees the predator, they're going to either run or hide, right? That is in response to what's going on in their environment. The predator sees the prey, it's going to chase, right? When I go outside and it's super sunny and I have been inside for a long time, then I'm going to squint, right? Because the light to my eyes causes me to squint. And further, my pupils, right, are going to constrict to limit the amount of light that's coming in. So we can think of a lot of examples for responding to the environment, or we could say responding to stimuli, right? Those would be the same thing. Whoops, that's supposed to be an I. Okay, reproduction That one's pretty straightforward, right? Now, when we think of, we probably typically think about humans or animals as far as reproduction goes, but in small single-celled organisms, one cell can divide into two, and that is also reproduction. Now, after reproduction, we have growth and development. Again, that's easier probably to think of examples with animals or with plants, right? Plants have growth and development. They start from a seed, and they grow in size and they develop, right? Flowering plants will develop flowers, or non-flowering plants will develop pollen, or not pollen, excuse me. Flowering plants will develop pollen. Others, like conifers, will develop naked seeds or cones. So those are also examples of growth and development. Now, if we think about another one that's very important, okay, homeostasis, or maintaining a balance. So that can be for us because we're able to control our body temperature, right? That our body uses its metabolism to help control our internal body temperature. Now, also, we control many things like the glucose levels in our blood. It is controlled to keep everything in balance. Our breathing rate, right, is controlled. If I go start running, it's going to increase my respiration rate so that I can get more oxygen to my cells. So there's a lot of things that are happening to keep a balanced level in a living organism. Okay, let's also talk about energy processing. Whoops. So if I stop eating, right, it's going to take a few weeks or a month, but I'm going to die because I don't have energy. My body needs energy to do its functions to carry it out. So whether it's a single-celled organism that needs to take in a food source from the environment, or a complex animal like us, we all have to take in an energy source like food and then convert it into something our cells can use for energy. So later in the semester, we'll talk a lot about how that happens in our cells, but all organisms have to, in some way, process energy. Now let's talk for just a second about a couple of things that aren't required for living things. So for example, we didn't say anything about movement. So you don't have to be able to move, right, to be alive. There are lots of single-celled organisms that don't have the ability for movement, right? Plants, now, you could argue that they can grow toward a light source, right? Or their seeds can get wind dispersed, but not movement as locomotion, okay? The other thing that we did not mention that's not necessary would be communication. Now, that doesn't mean that many living organisms don't have communication. Of course, we know that they do, but it's not a necessary, right, requirement. Not all living things have the ability to communicate. So that's just a couple of examples that aren't characteristics of life. So I believe I'm going to just move on to lesson 2, but before I do that, I'm going to mention one last thing that was touched on in lesson 1, and that is that everything living is organized into 3 domains of life. So the domains are eukarya, bacteria, and archaea. Now, we're going to talk later into lesson 4 more about cells in particular, but I do want you to know that everything living goes into these 3 categories, and there's some very basic characteristics that you should know, and the first is that eukarya is made up of organisms that are made up of eukaryotic cells. So everything in eukarya is made up of eukaryotic cells, and what all does that include? Well, it includes 4 kingdoms. So Animalia, which would be animals, Plantae, which is plants, Protists, which if you take 1409, you'll get to learn more about protists, and then fungi. Okay, so all of those four kingdoms belong in domain Eukarya. Now, bacteria and archaea are both made of prokaryotic cells. So again, we'll talk about the difference between these a little bit later. And these are all single-celled organisms. Okay. Now, the interesting thing about archaea, we can just say extreme. Typically, they're going to live in extreme environments. But not known to be pathogenic to us, whereas bacteria, most bacteria are helpful, right? We have more bacteria living on us than we have our own cells, and they're typically helpful, but we also know that there are pathogenic bacteria that we have to deal with. Okay, so let's move on to the chemistry chapter. So I'm just going to hit the high points. Let me know if you've got questions, or you can look back at the videos and the lessons. But up here in the top left, change my color, is an example of an atom. Okay. And what do I need to know about an atom? Well, every element in the periodic table is made up of atoms. So an atom is sort of the smallest particle that's still that element. So if you broke it down further, it would no longer be that element. And an atom is always made up of three subatomic particles. Okay, the proton, the electron, and the neutron. The proton is positively charged, the electron is negatively charged, and the neutron doesn't have any kind of electrical charge. And there's important things we should know about each of those subatomic particles. Okay, in the very center here, this is called the nucleus of the atom, which shouldn't be confused with the nucleus of a cell, right? And the nucleus of a cell, we'll talk about later, has the DNA. In an atom, this is very, very small, right? Much smaller than a cell. The nucleus just means that is where the protons and electrons are, I'm sorry, the protons and the neutrons are found. So right here in this nucleus, this contains the protons plus the neutrons. So protons are positively charged, neutrons don't have a charge. Now in orbit around the atom are the electrons. So we're just going to say they're always moving. Okay, so they're in orbit around the atom. Now we can use the periodic table to tell us how many protons, how many electrons, and how many neutrons an atom has. And we'll look at the periodic table in a minute. This is an oxygen atom. So if you have a periodic table handy and you want to look at it while we're talking about this, that you certainly can. If you want to look at the periodic, let me see, actually I can't bring them both up at the same time. So I'll show it to you in just a second. But if you look at the periodic table, let me find this one real quick. And you look at oxygen, what's important is there are two numbers. So if you can do that, go ahead and look at oxygen where you're at. In the little box for oxygen, I'll draw here what it looks like. There's a number 8 on top, and there's a number, it either says 16 or it says 15.999. So I'm going to just draw here what the box for oxygen looks like. So it has an O, right? And there's an 8 up here, and I'm going to put 16 down here. It depending on your periodic table, it may say 15.999. Okay, but we're going to round that to 16. We're not going to worry about the decimal place. Okay, the number on top is called the atomic number, and it is always equal to the number of protons. Now the bottom number is called the atomic mass, and it is equal to the protons plus the neutrons. So what that tells me is an atom of oxygen always has 8 protons, and its mass is 16. Therefore, if there's 8 protons, then there must also be 8 neutrons, right? You just add them together to get the total. Now, these numbers aren't telling me how many electrons specifically, but for any electrically neutral atom, so that's all we're going to deal with. If you go into chemistry, you talk about more complicated things, but for now, for us, the protons and electrons are balanced, right? Until we start talking about giving and taking for bonding. So meaning if there's 8 protons, then there's going to be 8 electrons. And just recall from the lecture that the electrons, we talk about where they're most likely. We know they're in motion, right? But we know that they exist in different energy orbitals. So we're going to talk about where they're most likely to exist. And they go into what we call shells. And this first shell will only hold 2 electrons. So you can see here's an electron here, and here's one there. And the next shell can hold up to 8 electrons. So you can see that we only had 8 electrons total, so 2 went into this first shell, which only left 6, right? 1, 2, 3, 4, 5, 6 for the second shell. So that means there's basically 2 empty spots in that shell. We call the outer shell the valence shell. So in this case, it's the second shell. If it had more electrons and there was a third shell, then the third shell would be the valence shell. But we call that outer shell the valence shell, and basically the way you look at this is atoms always want their valence shell to be full, okay, with electrons. So we have 2 empty spots. So that means this atom is going to be seeking to bond in order to make those full and not empty. I thought of something I just want to go back and say. I don't know why it just came to me. I was talking about something and I remembered. I don't know if I said this previously as we were going from atom all the way to biosphere, but I just wanted to reiterate that where something is considered living is at the cell. So if it goes atom, molecule, organelle, it's the cell, right, where things, we consider them living. So anyway, that's just review. Okay, so we have protons, electrons, neutrons. We are going to be focused on electrons when it comes to bonding. So if I move over to this picture right here, this is showing us about ionic bonding, and we'll talk about that. Down here is bonding, and this is covalent bonding. Okay, now bonding deals with electrons, so just keep that in mind. Remember, on the periodic table, the atomic number up here is the same as the number of bonds. So I just want to make sure that I explain. An oxygen atom always has 8 protons. It can never have a different number of protons, because if it did, it wouldn't be oxygen anymore, right? It would be a different element on the periodic table. So if oxygen doesn't gain or lose protons, so if you're looking at your periodic table, you see that nitrogen has 7 protons and always has 7, and fluorine has 9 protons and always has 9. So I just want to make that clear that atoms don't gain or lose protons, because then it wouldn't be oxygen anymore. But atoms can gain or lose electrons, and we'll talk about that as it relates to bonding. The other thing I want to mention to you is the neutrons. So in your lesson, it talked about something called an isotope. Okay, and maybe you think of isotopes as like radioactive isotopes or dangerous, but all an isotope means is a different form of that particular element or atom. So this right here, the number 16, is the mass of the average of all the oxygen atoms on Earth. Okay, so there could be some oxygen atoms that actually have a little bit different mass than that, a little bit more, a little bit less. And that has to do with the fact that they don't have the same number of neutrons. So, for example, carbon, the normal atomic mass of carbon is 12. Okay, or when you look at the average, right, the majority of the carbon atoms are going to have a mass of 12. But there is a form of carbon that's called carbon 14, and that just means that in fact its mass is 14. And it has a greater mass simply because it has extra neutrons, right? So that it's still carbon, it's just an isotope because it has a different number of neutrons. So I just want us to keep these particles straight. Protons is the same as the atomic number, and that doesn't change. Neutrons can change, right? You can have different number of neutrons. That just makes a different isotope for that particular element. And electrons are what we're going to talk about as far as bonding goes. So I'm going to get rid of this so we can have a clean slate. Now, if we look over here, this is an example of ionic bonding where sodium and chloride, okay? Ionic bonding means electrons, so that's my shorthand for electron, E minus, because they're negatively charged. Electrons, okay, are going to give or take, right, in order to form a bond. So these little gray circles represent the electrons. So remember, the first shell only holds 2, so we've got 2 there. The second shell is full with 8. Okay, we've got 8 there. But sodium, if you look in the periodic table, actually has 11 electrons, so it has an atomic number of 11. Remember, if there's 11 protons, so 11 pluses, there's got to be 11 minuses to balance that out, right? So that means there's 1 electron in the last shell. Remember, the last shell is called the valence. Now, sodium is no different than other atoms. It wants a full valence. So it could either gain 7, right, which is not real likely, or it could get rid of 1. If it gets rid of that 1 electron, then it has a full valence, right? 8 is in that valence. So it is going to be very motivated, okay, to give up an electron. Now let's look at chlorine. Now, if you have your periodic table handy, if you look it up, you see that chlorine has 17 electrons. So we're going to put 2 in the first, right, and then 8. And then this one only has 7, right, because there's 17 electrons. So there's 1 empty spot right here. So chlorine is highly motivated, right, to accept 1 electron. And so that's what's going to happen. Sodium is happy to give up an electron. Chlorine is happy to take it, so we'll add it here, and we'll get rid of it there. And ultimately what that means is we've created 2 ions. Okay? An ion just means when there is actually a charge. So the atom is no longer electrically neutral, it carries a charge. So in this case, sodium still has the same 11 protons, right? We didn't do anything with the protons. It started with 11 electrons, but now we only have 10 electrons because which are negatively charged, right? Because we gave 1 chlorine. So 11 pluses minus 10 minuses means it's going to have a charge of +1. Okay, so we'll write it sodium plus. Chlorine had 17 protons to start with. Okay, it had 17 electrons to start with, but it gained 1, so now it has 18 electrons or 18 minuses, which means it's going to have a charge of -1. So we would write it Cl minus. It is the attraction of these opposite charges that is what forms the ionic bond. So we would write that as just sodium chloride because the plus and minus charges, right, are going to cancel each other out basically. So that is the definition of what an ionic bond is, and it's just formed between 2 ions because one atom is giving and one's taking an electron. Now let's look down here at the covalent example. And I've used water, okay? Covalent means that the electrons are not given or taken, they're shared between the atoms. And when atoms share electrons, they actually both get to count the electron in valence count. Okay, so if you look up oxygen again, remember it has 8 total electrons. So here's 2, right, that go in that. Okay, so I'm sorry, this is oxygen right here. And these are hydrogen, so H2O, right? Oxygen has 8, so 2 fill that first valence, which leaves 6 for the next shell. So this one, this one, this one, this one, this one, that's 4, this one, and this one, okay? Now, hydrogen only has 1 electron. I'm going to color it a different color here so we can see what we're doing. So hydrogen has 1 electron there, and this hydrogen has 1 electron. So because they're sharing these 2, okay, in this bond, and they're sharing these 2 in this bond, they both count those in their valence. So since hydrogen only has the first shell, it's full with 2, right? So its valence is full. And oxygen gets to count 1, 2, 3, 4, 5, 6, 7, 8. So oxygen is full and happy. So that is the covalent bond is a stronger bond than an ionic bond. Now, covalent bonds can either be polar, which basically means they're not sharing equally, or it can be nonpolar, which means they are sharing fairly equally. Now, I did have a question about electronegativity. What is electronegativity? Electronegativity means that is how much an atom is going to be attracting electrons. So how much is the pull it has on the electrons? So if you think about it with, like, if you've ever seen people playing tug of war, if you think about that knot in the middle of the rope as the electron, and you have 2 players, right, trying to play tug of war, that's like electronegativity. How much is that atom pulling on that electron? So if they have equal pull, right, about the same amount of pull, it's going to be a nonpolar bond. They're shared fairly equally. But in the case of water, it is not equal. Oxygen has a much higher electronegativity than hydrogen does. Therefore, those electrons in that bond are not going to be shared equally. They're going to spend much more time around oxygen than they are around hydrogen. And that leads to some really interesting properties of water, because water, okay, is polar, which leads to hydrogen bonding, which leads to really interesting properties, like the fact that ice is less dense than liquid water. So the solid form is less dense than liquid water, which is not a common thing, right? Now, you can see that you might read this, that it says really positive. So when you look at an atom of, a molecule of water, it's going, let me clear this out so we can see this better. So if you look at an atom of a molecule of water, where the end that's closer to the oxygen is going to be, it's not charged, it's not an ion, it's weakly negative, it's like partially charged. Whereas the end down here by the hydrogens is partially positively charged, like weakly positive, right? Not completely charged. So what that means is different water molecules are going to be attracted. So the hydrogen right here in this water molecule, this end that's more positive, is going to be attracted to the oxygen in another water molecule, right? And the oxygen in this water molecule is going to be attracted to the hydrogen of another water molecule. That's all a hydrogen bond is. So if this is attracted to a hydrogen, okay, that's bonded to another water molecule, that right there is called a hydrogen bond. And this is what gives water some of its really unique characteristics. So if you think about cohesion, that means water molecules want to stick together. Why are they sticking together? Because of hydrogen bonding. If we think about why does ice float on water, that's simply because as water is cooled down, right, molecules are always in motion, and as you cool something down, they slow down. And when they slow down enough, every water molecule is going to be hydrogen bonded to 4 other water molecules. In order to make that happen, it has to spread the water molecules out into this lattice-like structure, which means there's more empty space in between the molecules. So in other words, you can just fit less water molecules into a given space when it's ice than you can liquid water. It makes them lighter, right? Less dense, so they're going to float. So I know that we have limited time, so I'm not going to spend too much time, but you should review all the characteristics that make water interesting, besides just cohesion and hydrogen bonding and ice and all that good stuff. So those of you that are here tonight, feel free to type in a question in the chat. I'm going to try to look back and make sure I cover those that have been sent in to the Google form. But otherwise, I'll just keep going. If you have a topic you want me to cover, then just let me know. I'm just going to peek back at the Google form real quick. Make sure I'm covering everything. Okay, I have some questions about scientific methods, so we covered that. Some questions about how do cells reproduce? No, you don't need to know that yet. We'll get there. Do we need to know the phylogenetic tree? No, you do not. You just need to know the 3 domains, right? What we talked about a little bit ago, so that's on the recording. 3 domains and some of their basic characteristics. We covered electronegativity. Okay, so it says on the study guide, the question about an atom gaining an electron and its resulting charge and what type of bond is most likely. So we just talked about that, so let's review. If an atom gains an electron, then it's gaining a negative, meaning it's going to be negatively charged. And if an atom is, that forms an ion, right? If it gains a charge, it's going to be an ion, which means ionic bond. Because an ionic bond just means attraction of opposite charges. So hopefully that answered the question of the person that wrote in. If not, email me and I'll try to do better. Why are some covalent bonds polar and nonpolar? That's simply to do with whether or not they have sort of equal electronegativity, if they have equal pull on the bonds or not. If not, they're not going to share very nicely. Okay, this next question I haven't covered yet. Sounds like a good time for us to do it, and it is about pH, hydrogen ion, what happens to pH. So let's cover that quickly now. I'm going to just get me a whiteboard. Okay, so first thing we're going to do is draw the pH scale. So we have 14 up here, and we have 7 here. Okay, so 7 would be a neutral pH. The higher we go up to 14, okay, it becomes more and more basic. And below 7, the lower the number, the more acidic it becomes. Now, what does that mean, basic and acidic? Okay, essentially, we know that this is water, right, H2O. Well, it turns out that water can break apart into ions. So this ion right here is called a hydrogen ion. And this one, okay, that's an OH. I don't know if that's clear. OH minus. That's called a hydroxide ion. there are certain things, when added to water, are going to increase the amount of hydrogen ion, and there are some things that will decrease the amount of hydrogen ion. So if you're decreasing the amount of hydrogen ion, then you're increasing the amount of hydroxide, right? So as you go from neutral to more acidic, that means you get higher and higher hydrogen ion, and this bracket means concentration. So in other words, the lower the number, the higher the hydrogen ion concentration becomes. Now, the opposite is true. Okay, when the pH becomes higher and higher, you're having an increase in the hydroxide ion concentration. Okay, so let me go back and see what her exact question was, or his, I'm not sure. An increase in the concentration or amount of hydrogen ion. She wants to know what that does to the pH. So an increase in the amount of hydrogen ion concentration lowers the pH, that's correct, which makes it more acidic. A decrease in the amount of hydrogen ion raises the pH and makes it more basic. So that's sort of a quick overview of pH. Okay. Okay, why was Chapter 3 not split into 2 lessons? There was a great deal of confusing information being presented that might have been easier to tackle if it was split. So, yes, Lesson 3 has a lot of information. Unfortunately, though, I have to get everything into the 16-week semester, so in order for us to get through everything, we have to get it covered like this. So I'm going to go over Lesson 3, some of the highlights, and hopefully it isn't as confusing after that. Okay, do we need to be able to differentiate and recognize the structure of the functional groups, or just know what they do and how they are formed? So I'll talk about what you need to know about functional groups. And then there's some questions about polysaccharides. So I will make sure to cover these. Okay. So that sort of leads us, oh, you know what, I just wanted to mention to you, down here in the bottom corner, I have some information about carbon. So remember, if something is organic, that just means it contains carbon and hydrogen, right? Specifically carbon. And carbon is, we're carbon-based, right? Our proteins, our nucleic acids, our lipids, most of the molecules in our cells are all made out of certain particular atoms, carbon being one of them. So carbon, hydrogen, oxygen, and nitrogen, or CHON, are the 4 most common atoms in living things. We also have sulfur and phosphorus, right? Because we know that those are found in our proteins and in our DNA. But carbon is outstanding in that every carbon atom can make 4 bonds, right? It's making 4 bonds. And it has the ability to make chains and make rings, and there are, as mentioned previously, functional groups that can be added on that change the chemistry. So carbon is, if you're talking about building blocks, right, it's got unlimited potential. Okay, I'll get back to functional groups in just a second. Let's move on to macromolecules. So sometimes these are called biological molecules. Essentially, these are the big molecules of the cell. And there's 4 groups, okay? Carbohydrates, lipids, proteins, and nucleic acids. Now, I'm going to put a circle around lipids because it's a little bit different than the other 3. But for the most part, these other 3, and then part of the lipids, They have monomers and they have polymers, okay? And a polymer just means you put a bunch of monomers together, right? You stick a bunch of monomers together and you end up with a polymer. So you should definitely know what the monomer and polymer or polymers are for each of these groups. So we can talk through that. Now, here's some thinking questions that are going to tie in with macromolecules. You know, I'm not understanding exactly how this relates to me, or it just seems abstract, right? So you eat, right? Let's say you have a big meal. I wish I had that pasta right now. And right after you eat, the blood glucose levels go way up, right? Which, well, not way up, but they begin to increase. And what is going to happen, because it's not healthy, right, for your blood glucose to get too high? So what's going to happen to keep it from getting too high? And so, well, somehow that glucose has to get stored away, right? So glucose is a single sugar molecule, and the glucose actually gets these single sugars are a monomer of a carbohydrate, right? It's the individual building block, it's the monomer. And these glucose are going to get bonded together to form a polymer. And the polymer that for animals, for us, that gets built up is called glycogen. So glycogen is many, many glucose, right? So this would be a polymer. Those glucoses get bonded together into glycogen, and then it gets stored away in your muscle cells and in your liver until later when your blood glucose begins to drop because you haven't eaten in a while. And the glycogen then can be released and broken down into individual glucose molecules so that your body can use that for fuel. Now let's talk about what are the monomers and the polymers of carbohydrates. Well, the monomer is called a monosaccharide. Mono means one and saccharide means sugar. So a single sugar. So we talked about one. Glucose is an example. Okay. And another one that you learned about in one of your assignments was fructose, right? Fructose is also a single sugar, and it is what causes that sweet taste, right? So those are two individual monomers. Okay, the polymer is called a polysaccharide, and there's four that I want you to know: starch, glycogen, which we just mentioned, chitin, and cellulose. Now, starch, we know that certain foods are high starch foods, right? That's because plants store their excess glucose as starch. So we store ours as glycogen, they store theirs as starch. So those are both energy storage forms of a polysaccharide. Now, chitin and cellulose aren't energy storage forms. They're actually structural polysaccharides. So cellulose is found in plant cell walls. It helps the cell wall have some structure to it. Chitin is actually found in fungi, and it's also found in the exoskeleton of insects. So again, this is a structural polysaccharide, not an energy storage one. Now, I have this picture of Legos because I want you to think about monosaccharides as being building blocks. So each individual Lego brick, right, is a monomer. And when you put a bunch of Lego bricks together, you've created a polymer. And so essentially there is a chemical reaction, right, that's going to do that, to go from monomer to polymer. So if we have individual monomers over here, and over here we have a polymer, the type of reaction that's going to do that in the cell is called a dehydration reaction or dehydration synthesis. And the reason it's called dehydration is because when those individual monomers get bonded together, there's actually a molecule of water that is released. So that's where the dehydration part comes from. Now the cell can also go the other way, right? From polymer to monomer, breaking a polymer down into its individual units, which is what happens when we eat our food, right? And it gets digested. So that is called a hydrolysis when it's the other way, when you're breaking them down. That's called a hydrolysis reaction in the cell. Now, there's another term that you read about, and or two terms that mean this. And so there's anabolic and there's catabolic. Okay, so if you think about, if you've probably heard of anabolic steroids. Okay, so we think about that like they're using those so they can build up muscle, right? An anabolic reaction is when you're taking the monomers and you're putting them together to build the polymer. Okay, a catabolic reaction is when you're going from the polymer and breaking it into individual monomers. Now, we talked about carbohydrates, right? We talked about the monomer is monosaccharide and the polymer is a polysaccharide. I do want us to look at what, here we go. So this is just a bit blurry, but notice that this is a structure of glucose and this is a structure of fructose. Those are the monomers, right? The monosaccharides that we talked about. And notice that basically they are ring structures, okay? So one of these rings, right, would be a single sugar or a monosaccharide. Now notice down here, this is sucrose, which is a disaccharide. Di means two. Notice that now these two sugars are bonded together. So that would be one reaction has happened, right, to form a bond between those two sugars. Now to build up a polymer, there would be multiple, right, of these happening so that you would have many, many sugars bonded together. But that is basically the structure of a monosaccharide. It looks like this six-membered ring. Now we want to look at the polymer. What does the polymer look like? Okay, so each one of these, right, is one glucose, okay, in these structures of polysaccharides. So these are some examples. So we talked about glycogen and we talked about cellulose. So notice it's many, many, many glucose molecules bonded together. Glycogen is branched, whereas cellulose is sort of more in a more ordered fashion. Okay, let's move on to talk about the monomer of a protein. So a protein, you have many proteins in your body doing lots of different things, and the protein itself is the polymer. Okay, it's made up of amino acids. So I'm going to put the amino acid is the monomer. Okay, and this structure here, this great gigantic structure, this is one single amino acid. Now, this carbon in the middle is the same for all. There are 20 total amino acids that exist. This carbon right here that we call a central carbon, it's the same for all 20. This hydrogen's the same. This group right here, which is called an amino group, is the same. This group, which is called a carboxyl group, is the same. The only thing that's different is right here, where it says R. So R means this is the variable group, right? That can be different. So it could be as simple as a hydrogen there, or there are complex amino acids that have ring structures where that R group is. But each of those 20 amino acids is one monomer of a protein. And so the protein will have hundreds of amino acids, right? Thousands of amino acids bonded together. And it's the order in which the amino acids get bonded together that create the specific protein. And we'll learn, we'll talk about it even more later, but the instructions for how that protein is to be made, in other words, which amino acid comes first, which amino acid comes second, the instructions for that are instructed for in your DNA. So for every protein, there's a gene that says this is the order that the amino acids need to be added. So while I'm on this page, I want to go ahead and talk about functional groups. So someone had sent in a question like, what do we need to know? So there's a few functional groups that I want you to know. Two of them are on an amino acid. So this group right here, oops, just a second. This group right here that has a nitrogen and two hydrogens, it's called an amino group. That's where the amino acid is named from. So I want you to be able to recognize that if there's a nitrogen and a couple hydrogens, then you know that's an amino group. This one here, okay, is called a carboxyl group. So it's a carbon. That means a double bond, right, to an oxygen, and then it's got a bond to another oxygen and hydrogen. Carboxyl group. Now I'm not going to try to trick you by putting, a carbon and double bonded to oxygen and then double bonded to another oxygen or something. If I showed you, an amino group and a carboxyl group, I would like you to be able to identify this one as the amino group. Okay. Now there's a couple others that I want you to know. Let me just get the white. Actually, I can just draw it here. It's fine. So just an OH, okay? Like if this were just by itself off of a carbon, this is called a hydroxyl group. Okay, so I want you to know that one. And I think that's probably, oh no, I'm sorry. I need you to know to be able to recognize a phosphate group, because we're gonna talk about phosphate quite a bit as it relates to DNA as we go on and talk about genetics. And so a phosphate group essentially has a phosphorus. Let me just clear this out. Okay. Whoops. So a phosphate has a phosphorus in the middle. It's going to be double bonded to an oxygen, and it's going to have other oxygens around it. Okay. Some of those oxygens are going to have hydrogens on them. I'm not interested in whether it has hydrogens or not. If there's a phosphorus with 4 oxygens around it, okay, in any way that the hydrogens are, that is a phosphate. So you've got carboxyl, amino, hydroxyl, and phosphate. So as long as you don't have to draw them or anything, but just to be able to recognize those groups is going to be good. Okay, let's see here. Now, I think I'm about ready to move on to the cells, but I did want to mention, so we talked about carbohydrates, we talked about the monomers and polymers, we talked about proteins, and the monomer is an amino acid, right? The polymer is a protein. With nucleic acid, I should probably cover that just briefly. So with nucleic acid, there's DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Those are the big polymers, right? They're all made of nucleotides, okay? So this right here is one nucleotide, and this is the monomer of nucleic acid. So remember, what's the polymer of nucleic acid? It's DNA, deoxyribonucleic acid, or RNA. And again, we're going to get into more details about that later in the semester. But what do I mean? What is a nucleotide exactly? It has 3 parts. It has a base, specifically that contains nitrogen. It has a sugar, and it has a phosphate. So we just talked about the phosphate group. So what do I mean there? Okay, this part right here, that's the sugar, right? You see that ring structure? This part right here, that's the phosphate, right? The phosphorus with the 4 oxygens around it, that's the phosphate group. Now, this part right here where it just says base, what that is, this part changes. So there are 4 letters in the DNA alphabet, and there's 4 letters in the RNA alphabet. So this is the only part that changes the base. So for DNA, that base can be A, C, G, and T. And for RNA, everything is the same except there's no T, there's U. All right? So you can see over here, this is the chemical structure for each of those. So where it says base, one of these would be in its place. So if this particular one was there, it would be an adenine, pardon me, an adenine nucleotide. So the nucleotide is the monomer. When you put a whole bunch of those nucleotides together and they're bonded together, that's when you have the polymer of either DNA or RNA. Okay. Now to move on and talk for just a little bit about our chapter this week, or our lesson this week, which is on the cell. So the main thing to keep in mind, we talked about the 3 domains, and there are only 2 basic types of cells. There are prokaryotic cells, and there are eukaryotic cells. And the main difference between those, prokaryotic cells have no nucleus. Eukaryotic cells have a nucleus. And when I say nucleus, it could also mean other organelles. Okay, so prokaryotic cells have no organelles, no nucleus, whereas eukaryotic cells have a nucleus and they have other organelles, or can have other organelles too. Okay, typically eukaryotic cells are going to be larger, they're going to be more complex, whereas prokaryotic cells are smaller, right, and simpler. Now, I have, pardon me, an example prokaryotic cell up here, but before we talk about that, I want to talk about these 3 terms right here, because these are sometimes confusing and cause problems for students. So cell membrane, plasma membrane, and cell wall, let's talk about how all these relate to one another. Well, a cell membrane and a plasma membrane are the same thing. Okay, they're 2 terms for the same thing. They are the membrane that is basically separating what's inside the cell from what's outside the cell. So every living cell has a plasma membrane. Now, a cell wall is not the same as those, because first of all, not all cells have a cell wall. And even if cells do have a cell wall, they also have a cell membrane. So the cell wall is exterior to the cell membrane, and it is not necessary. Okay, you don't have to have a cell wall to be alive. Now, there are some things that all cells have in common, whether it's a prokaryotic cell or a eukaryotic cell. And those things that all cells have in common are the plasma membrane. I just mentioned that, right? Ribosomes, these are protein factories. Cytoplasm, which is the cell soup, okay? And DNA. Now remember, I said prokaryotic cells don't have a nucleus, and they don't, but they have to have DNA. The difference is the DNA, as you see it here in this red right here, is not in a membrane-bound organelle. It's just free in the cell. Now, notice that it does say nucleoid. That's not the same thing as a nucleus. That just means this is where the bacterial DNA is located. So you see it's a pretty simple cell. There's genetic material. We have ribosomes in the cell. We have cytoplasm in the cell. There is a plasma membrane. In this case, it shows a cell wall, which most bacteria do have a cell wall. Okay. Now, in this case, it shows a capsule. Some bacterial cells can have a capsule on the outside of the cell wall, and this is really important if you go on to take microbiology, when you talk about if a cell is pathogenic and how infectious it is versus how hard it is to treat it. That comes into play. The pili right, which can be used for bacterial cells to actually exchange information. And then, of course, some bacterial cells can have a flagella. They don't all, okay? They don't all have a flagellum, but some can, and this helps them with movement. Now, what's shown here is an animal cell, and then, of course, a plant cell. Now, most of the organelles inside of the animal and plant cell are going to be the same, but there are some differences, right? For one thing, a plant cell is going to have a cell wall, and an animal cell does not. A plant cell is going to have a chloroplast. This is really important for photosynthesis, right? This is how it harnesses sunlight. Animal cells don't have chloroplasts. The other thing that a plant cell has is a central vacuole. So in this case, it's going to fill with water and press out against the cell wall, which provides something called Turgor pressure. So if you forget to water your plant and it kind of wilts, that's because it's not exhibiting very much Turgor pressure, right? Those central vacuoles aren't very filled with water. Now for animal cells, let's talk about something called centrioles, okay, which are these little things right here. They play a role in cell division. So when we talk about cell division, you'll learn the details of that. But those centrioles help form the spindle fibers for cell division. Now, plant cells still divide, but they don't have centrioles. Okay. The other thing, or organelle that animal cells have, are called lysosomes, and these are basically digestive enzymes that are packaged up within a membrane that digest old cell parts or invaders or foreign particles, chew it up, recycle those parts. Now I'm going to just pause right here for just a second and ask if anyone that's live in the session has any questions that they want to put in the chat box. And I'm just going to take another quick look back at the Google form and make sure I've covered everything. So there were no questions sent in about lesson 4. So it may just be because that's so new. So what I will tell you is there's definitely some good videos to watch for lesson 4. So I highly recommend that you do that because you do need to know all of the organelles that are found in the cell and their functions, what they're doing. So when you go to lesson 4 lecture folder into the videos, I have a quick description there for you of like the cell theory, which is important to know that, you know, everything living is made of cells. The cell is the basic unit of life, meaning nothing smaller than a cell is considered living, and that cells come from cells. Contrary to what they thought hundreds of years ago with spontaneous generation, cells don't just come out of nowhere, right? And it goes on and talks about the 2 types of cells, and then there is a video that I've created that looks at animal cells and plant cells. So make sure you're watching those videos, looking at the study guides, to retake some of those quizzes right at the end of the lesson, see if you can go through some of those questions without looking at your notes. You can have a note card. If you have me in class, you can have a note card that you can use for Proctorio. It has to be handwritten. It can't be typed, but you can put information on that note card to use for the exam. Not your notes, not all the study guides, just a note card. I have noticed that there may be some issues with Proctorio and the instructions showing up, so I sent those out to everybody to make sure that you have seen those ahead of time. One thing that I've noticed that helps is you may have to clear the cache and clear your browsing history to make sure that everything will work correctly with Proctorio. So I really encourage you to go ahead and use that Try Proctorio link that's there in the units folder to make sure your computer's all set up and everything's working correctly with Proctorio before you go to take the exam. But with that, I'm just going to open it up to any other questions. So yeah, you've got a question, that's great. If you can type it in the chat box. So it may be that you don't see the chat box. If you don't see the chat box, there's a little kind of purplish pink thing with two arrows down at the bottom right corner. If you'll click on that, it'll open up and there'll be a little speech bubble that you can type a message in to me. If you hit that speech bubble. Or if you have audio, you can use your audio if you want to ask it. Or if that's not working, you send me an email real quick. Yes, so an index card size, yes. So exactly, the note card, and it doesn't, if you don't have index cards, that's okay. Cut a piece of paper, that's like 4 inches by 6 inches. So index cards come in several sizes. The normal size, the small ones are 3.5 by 5. That's fine. Or the little bit bigger size, the 4 by 6, that's fine too. So yeah, an index card works well. And other questions that you may have about the class or about lab or about grading or anything else, you're welcome to ask that. I'm just looking to see if there was anything I left out. I think we've covered everything I had planned. So I am, I guess, going to say good night to you. Thank you to those of you who joined me this evening. I appreciate you being here. I hope we will end up with more next time. Um, let's see. I will give both of you some bonus points for joining me tonight, and maybe that'll encourage others for next time. So I'm just going to write your names down super quick. All right. Any other questions?